Hello and welcome to the tutorial for i3CR Vano. In this tutorial, I will be walking you through the usage instructions of this framework using Docker. The source code and all pre-trained AI models of this framework are available at these two GitHub repositories, Energy AI Neuronco Preproc and Energy AI Neuronco Segment. And note that all the documentation for using this framework are present in the first repository, that is Energy AI Neuronco Preproc. Now let's move on to exploring the first repository and look at the documentation. So this is our Energy AI Neuronco preproc GitHub repo, and if we scroll down to the README file, we will see the usage instructions using Docker and XNAT. And in this video, we will be exploring the Docker option. So looking at the usage using Docker, I have the installation and requirements. So of course, to run it using Docker, you will need Docker on your setup. Then we need to pull these two Docker containers. The first one does everything except segmentation, and the second one does the segmentation. And then you have to arrange all your data in the given input folder structure, and the commands for that are given in this documentation. Once everything is set up, you have to run these 6 commands as they are given in this documentation to process the data from start to end. Now let's look at an example. So we have a sample session TCGA020047, inside which we have 10 different scans of a patient, and notice how the scan names have space in them. So the first thing we do is we check if the Docker images are present. And we see that the two images are present in our setup. And so the next thing we do is we arrange, we check the folder structure and remove all the space from the names of the directory and all the file names recursively and replace them with underscores. Next, we set the input path and the output path variables. For the input path, we set it as the path where all the scan directories are present. And for the output path, you can set it as the path where you want all the outputs to be stored. And so in this case, we set the output path as this, and we create all the required directories and subdirectories. All these commands are taken from the GitHub repository directly, so you can just copy paste them. So once all the directories are created, now we run the first command of scan type classification, and scan type classification runs. So now the scan type classification has stopped running, and we can see the classified predictions of each scan, and we can also see that the segmentation ready flag is set to be true. That means this session has all the required scans needed for segmentation. Inside the output directory, we can see that it has created the 4 scans that we will need for downstream processing. And having finished that, we move on to the registration step and run the registration. So the registration is running now, and I have sped up this processing for the sake of this tutorial. And it took about 4 minutes to run this registration. And we go inside the to registration output directory, and we see that inside the co-registered folder we have the co-registered scans, and inside the registered folder we have the registered scans. Next, we run the skull stripping. The skull stripping took about 1 minute, and it created all these underscore stripped files within the skull strip directory. And the next step is segmentation. So in segmentation, you can have the evaluate and the radiomics flags turned on, or you can set them off. So I have them set as on. The segmentation is running now. And we also see the radiomic features being generated. Now the segmentation has finished running. We go inside this for segmentation directory and we see the radiomics.csv and the prediction files. In the next step, we convert this tumor mask from atlas space to patient space, and it is created in this segmentation patient space directory. And then we convert this nifty mask in patient space to DICOM seg or DICOM segmentation object format, and this can be used as a final tumor mask. And now we have finished running the entire framework, and let's look at the outputs. So first we have this scan type classification outputs. Inside the scan types directory, we have this prediction classifier meta with provenance.csv file, which contains the prediction information of all the scans present in this session. So we see that the I3CR Vano framework also generates the orientation information as well as the predicted scan type. For debugging purposes, we also have the predictions produced by these two classifiers individually. So the classifier 1 classifies them as anatomical or not, and the classifier 2 does a more granular. We have this reference column which is set to true for scan number 9. That means this scan is used for as a reference scan for all the co-registration purposes. And in the prediction column, you can see this dash seg suffix to some of the scans. That means those 4 scans will be used for all the downstream segmentation purposes. That is the scan numbers 4, 5, 9, and 10. These 4 scans of these 4 different sequences are selected for all downstream processing. We also have this acquisition parameters.csv file where all the DICOM acquisition information is stored for all these scans. We also have this anat patient space directory, inside which we have the 4 selected scans in nifty format, the T1 pre-contrast, post-contrast, T2, and these flare scans, as you can see. And from this image information, you can see that all these scans have different dimensions and different voxel spacing, and this will all be fixed in the next step, that is registration. This is the output of the co-registration, in which we have all the scans registered to the T1 contrast scan for this session. So we have this T1 contrast, then T1, then the T2 scan, and then finally the flare scan, and all co-registered to the T1 post-contrast scan. And if I look at the scan slice by slice, we can see that they are all aligned to the T1 contrast scan. Looking at the image information, we see that all of them have the same dimensions and voxel spacing. Now the registration step also performs a registration of all these co-registered scans to an standard atlas, and in this case all the co-registered scans are also registered to the SRI-24 atlas. So these are all the registered scans registered to the SRI-24 atlas, and if we look at the image information, we see that they have all dimensions of 240 cross 240 cross 155 with a voxel spacing of 1. We also have this qc.png file for quick visualization of the registration outputs. So if something is wrong in the registration, you will be able to see from this qc file. Next, we have similar outputs for the skull stripped outputs as well, and I have sped up this process because these outputs are very similar to registration. We also have a QC file for this. And next, moving on to the segmentation output, we have this prediction.nifty file, which is the multi-class tumor mask generated by the segmentation model. And we can overlay this mask on these registered scans to visualize the tumor. So here I have the T1C and the FLAIR scans, and I have overlaid this tumor prediction on these two scans in the ITK snap tool. And you can see this multi-class tumor mask. The green is for the edema class for the glioma. The yellow part is the necrotic core. The yellow part is the enhancing core, and the red part is the necrotic core of the tumor. We also have this radiomix.csv file, and also this 2QC files seg.png, where you can quickly take a look if the segmentation looks correct or not. We also have the zoomed version of the same file, where we have a more zoomed in view of the tumor. Let's look at the radiomix.csv file now. So we have these different shape features, texture features, and first order features for all the different tumor compartments. So ED, for example, stands for edema, NC stands for the necrotic core, etc. And then we have these features for all the different sequences, that is for T1 contrast, T1, T2, and FLAIR. The shape features are the same for all four sequences because they don't depend on the image intensities, but all other features like the texture features are separate for example, the FLAIR scan, the T1 scan, T2 scan, and so on. So for example, here you see that this particular column is for the necrotic core of the tumor, for the FLAIR scan, and the first order feature and of the 10th percentile. And here we are showing another example feature. This is the necrotic core FLAIR GLCM feature. So now moving on to the next step, which is the segmentation patient space. Now this is the same tumor segmentation mask but in the patient space. So you will be able to overlay this on the co-registered scans now, because these co-registered scans are in the patient space. So I open up this T1 contrast and the FLAIR co-register scans, and I overlay this prediction in patient space, and you can see that they are showing the tumor in the patient space. And you can change the opacities of the individual tumor compartments using the ITK snap tool. And then finally, we move on to the final output, that is a prediction patient in the DICOM seg object format. And we can visualize this DICOM seg object in the 3D slicer tool. And so now we know that the target scan for this is the series 9 or the 9 scan. So what we do is we open up 3D slicer. So for visualizing this final DICOM segmentation object, we will need this reference scan and the object both. So as we know that the series 9 is the reference scan for this session, we open up this series 9 from this raw scan directory, and we also open up, so we open up this scan 9, we import it, and we also take this final tumor segmentation DICOM seg object from the IC3R Vano output. Now we can see that both of these are in 3D slicer, and so we open them up for visualization, and you can see that the two modalities are being shown as MR and seg. So this is the final tumor mask in DICOM segmentation object format, overlaid on the reference scan, that is series 9, the T1 post contrast scan, and it shows these tumor compartments, these 3 different tumor compartments of necrotic core, edema, and the enhancing part of the lesion. You can turn these individually on and off using this feature of 3D slicer and look at these tumor compartments. So for visualizing this in 3D Slicer, you will need this quantitative reporting plugin, which you can install for free, and then you will be able to visualize this DICOM segmentation object. So this brings us to the end of this tutorial. Thank you for watching, and you can find all the code at these two GitHub repositories. Thank you.